Okay, the third paper is by Chengjie Xiong, who is from the Knight Alzheimer's Disease Research Center Data Management and Statistics Core. And he will discuss racial differences in AD biomarkers, design and methods of future studies.
Okay, can everybody hear me or see my slides?
Yeah, we can see it. Yeah.
Okay, great. Good afternoon, morning, or evening, depending on where you are. So I'm going to switch here a little bit to talk about biomarkers in Alzheimer's disease. And I will focus on the possible racial differences in those biomarkers. And this is my disclosure. I will start with some literature review. There are some of those, but not a whole lot in terms of racial differences in Alzheimer's disease biomarkers. And I will then present basically some data from our Alzheimer's Disease Research Center at Washington University here in St. Louis. Both cross-sectional data, some are published, some are unpublished yet, some are in press. And then I will talk about the design and analytical issues in those analyses. And in the end, I will make some remarks. So AD biomarkers, I think probably for this audience, I don't really need to go much further with this slides. It has been recently proposed this biomarker research framework in Alzheimer's field called E, which is amyloid, T, which is tau, and M, which is neurodegeneration. So those are the biomarkers from this either the CSF or the MRI or the PET imaging, including both amyloid and tau imaging, and most recently also from the plasma biomarkers. Now, of course, many of those biomarkers try to target the neuropathology of the disease, so it's also appropriate to talk about the racial differences in the pathology. All right, so some of the literature, in all I'm talking about here, we're talking about African-American self-reports versus whites. They're also self-reported. And African-American have been found to have a lower level of CSF tau, total tau, and the phosphorate tau at 181. This finding was probably first published about, I think maybe 3-4 years ago, but since then have been at least replicated by 5 to 6 studies. And some finding about the PET amyloid imaging, which is less consistent. African-American had higher or lower or similar levels of PET amyloid uptake, depending on which study you're talking about. And also about the tau imaging, the PET imaging, African-American either had a similar or higher SUVR in different regions than the whites. So again, things about the racial differences in tau imaging are not entirely consistent in the literature, and the literature is very few at this point. Plasma biomarkers is even newer. It only occurred very recently in terms of the racial differences. Those studies and this here are very recent. They found no differences at all between African-American and whites. However, one of the studies which is in press now found plasma A-beta-42 was higher in African-American group than the non-Hispanic whites, and also the plasma A-beta-42-40 ratio was also high, but there was no difference found in plasma p-tau 181 and p-tau 231 and NFL. The pathology finding in terms of racial differences is also quite mixed, but it's pretty consistent in terms of African American with AD pathology. They tend to have more mixed mixed pathologies with others. That seems to be quite consistent. And in terms of amount of AD lesions, AD pathology, there are also inconsistent findings in terms of pathology. Next, I'm going to basically show you some of the data from WashU Alzheimer Center. Some are published, as I mentioned, some are not. This is one of the first reporting racial differences in CSF biomarkers, especially in total tau and p-tau. And this study is from about over 1,200 participants. About 170 of them are African Americans. All the others are non-Hispanic whites. From the same study, we also found that if people report the family history of dementia, African American tend to have lower hippocampal volume than the whites. Now, this is also a recent study from our center as well on the CSF soluble TREM2. It basically found African American have lower level of TREM2 and are also more likely to carry TREM2 coding variants. Another study, imaging study from our center found that the racial differences, well, in this situation, it's in the neurodegeneration marker, more specifically the cortical volumes from the MRI imaging. The social economic status actually mediate this relationship between race and the cortical volumes. Now, those are new data. Our center just last year, I think, went through all the legacy plasma samples we have, and we used the C2N Diagnostics Preceded AD platform, a well-established platform, to actually assay all those samples. And those are the CDR, a subset of it, CDR0, clinical dementia rating of 0, which basically means normal cognition. That's the subset I'm presenting here, and the demographics and the ApoE4 information here. Now, if we run some of those unadjusted analysis, just compare the two races, you can see there is a racial difference between African Americans and whites in plasma A-beta-42/40 ratio, and also in plasma p-tau 231. And the CSF finding is in CSF A-beta-42/40 ratio is also significant, which with African American with a higher level of both plasma and CSF, A-beta-42/40 ratio. In the adjusted analysis, you can see actually some of those differences seems to be even stronger. We now see some type of racial differences in plasma p-tau 181 and also 231, and with the African American having lower plasma p-tau in both locations. Now we look a little further. The sample size is not so big, but perhaps one of the largest we have in this field about plasma biomarkers, especially in African Americans. So we look at some of those possible two-way interactions, look at how the racial differences we just looked at could be modified by other factors. And I list all those two-way interactions. The p-value is kind of like either significant or showing some type of trend. And those are the p-values here in this slide indicating some of those factors, covariates, things like gender, things like education, age, and E4 could all potentially modify some of the racial differences we just looked at. So just some example here in plasma A beta 42/40 ratio. If you look at the race by gender, you can say perhaps, well, they both show racial differences, whichever gender you're talking about, but in the male, it seems to show a stronger, a bigger effect size than the female in terms of the racial differences. This slide shows the racial differences as a function of the education. So, well, for the plasma NFL, and you can tell there seems to be some type of interaction. Of course, we need a much bigger study to validate this. But if this is true, it will have very significant implications in terms of interpreting those racial differences. Basically, what you're able to see here is perhaps some places in the middle education in this graph, you don't see much differences, but if you go lower or higher education levels, and there is a racial differences, but the direction is actually opposite of each other. And notes here in plasma NFL, I just reported in those tables, we didn't see a racial differences by only looking for the main effects. So that's why it's actually very important when we run this type of analysis, when we have a comprehensive model to look into all those possible modification factors. Those are new data. Well, it's actually is in press as of now. It's probably the first longitudinal data on racial differences between African American and whites on the major Alzheimer's disease biomarkers. This slide gives you a sense of the degree of the longitudinal data. We have white and African American. The first number is the number of people with at least 2 time points. Typically, our study is following those subjects every 2 or 3 years in CSF and the imaging biomarkers. So that's the biggest sample size, the first number. 37 of them have longitudinal CSF data, or 47 have, I'm talking about African American, of course, our Caucasian, our whites are much bigger, about 10 times bigger than the sample size of African Americans. The PET, amyloid, I'm talking about here, about 47 African Americans have longitudinal PET amyloid imaging. About 70 African American have longitudinal MRI, and a much bigger number with longitudinal clinical and cognitive assessment. So not everybody in this cohort, in this study, have all those biomarkers. So I'm going to present some of your results in specific to each modality. For the CSF cohort, this slide shows you the basic demographics, the ApoE, and also the Hollins head index. And largely African American and whites are matched by those parameters, and this in terms of mean, but there are differences in BMI, in social economic status. Some spaghetti plot of the CSF biomarkers. This is the A beta 42. To everybody's surprise, pretty much if you look at the spaghetti plot, the whites seem to have a trend going down longitudinally. That's what we expect. On the other hand, African American seems to have a trend going up. So it's very, very interesting to look at those graphs. And also, this is A beta 40/40 ratio. They both are going down over time, but one is going faster, going down than the other. And this is CSF tau. and the p-tau at 181, and turning those spaghetti plots into numbers. So I'm reporting here the slope, which is the annual rate of change. So how much the increase or decrease in those biomarkers for each race over a year. So as you can tell, the African American for A beta 42 over time goes up and the whites go down. So that resulting that the significant differences in terms of rate of change. The A beta 42/40 ratio also is significant in terms of the difference between African American and whites. And for total tau and p-tau from the CSF, the p-value is certainly not significant, but if you look at the estimated slope, the annual rate of change, African Americans' rate of change is about half of the whites. So this could very well be a power issue here in terms of the racial differences in the longitudinal change between African American and Caucasian in total tau and phosphorate tau. We're turning into the PET amyloid cohort. This is the all, again, as I said, those are all CDR0 at baseline. They're all normal at baseline. Again, the BMI and socioeconomic indicator index seems to be very different than others. Education is also maybe different. Others are quite similar in terms of the demographics and the ApoE. And the MRI cohort, that's a bigger cohort, of course. We have longer follow-up and more bigger sample size for MRI scans, and that's the demographic at the baseline. And the follow-up, the next follow-up is also a little bit different in the MRI, about 4.6 years for African American, about 6 years for the whites. So for the imaging biomarkers, those are some of the spaghetti plots. This is amyloid, PET amyloid in the Sentinel scale, simply because we actually have used over time the PAVE tracer and also the AV45 tracer. So we combine them into some type of Sentinel scale. And the hippocampal volume, that's the total hippocampal volume between African American and whites. And the cortical thickness. And again, turning everything, the imaging markers into the annual rate of change. And we can see there is a difference between African American and the whites in terms of the amyloid accumulation per year. The amyloid, the rate for African Americans is about half of that of the whites. And that's a significant difference. And all the other biomarkers from the MRI does not seem to differ in terms of their change. So we also look at possible effect of E4, ApoE4 on longitudinal racial differences we just looked into. So this give you the annual rate of change for each of the cells classified by both race and the E4, which is classified as positive or negative. And in the bottom two rows there, you can see the racial differences for either E4 negative, without an E4 allele, or E4 positive. It seems to be the racial difference primarily occur in the E4 negative, with African American having higher level of the CSF A beta 42. Looking the similar effect of the E4, but in the PET amyloid in the Sentinel scale, and this seems to be on the other way around. The main racial differences seem to occur in the ApoE4 positive subgroup, but perhaps not in the E4 negative group, based on this limited data set. So with all those, I wanted to turn to some of the design and analytical issues in those type of studies. from the published studies, the literature we just reviewed, pretty much all are based on convenience samples. None of those are really population or probability samples. All are retrospective studies, although some study reprocess all their samples, CSF samples, plasma samples, and imaging scans in a standardized way. Some didn't. Almost all are cross-sectional data. The data I just showed you seems to be the first in terms of racial differences in AD biomarkers about those longitudinal differences, and all have a pretty small n, especially in the group of African Americans. So those are the weaknesses of the published studies. If you look into the analytical approaches, there are some of those issues about assay forms that people are using, especially for the plasma biomarkers. I just presented several studies, found nothing. Using some different type of plasma assays. And one of the study, which I was involved, used the perceived AD from C to N that found some of the racial differences, which simply means perhaps the assay forms also matter. Almost all those studies focus on the main effect of race. Very few folks looked into the other factors in a comprehensive way. Of course, a small sample size limits those approaches. And even for some, the adjusted analysis ignored possible modification effect by covariates or confounding factors. And what we typically have done is some main effect of the covariates in this adjusted analysis. A very important issue here, of course, in any of those studies is the social determinant of health. None of those studies have consistently connected those data, and those analyses in the literature have largely ignored the SDOH in the analysis. Looking forward, what I think the field really needs, we certainly need population-based large biomarker studies. We're going to need standard assessment of social determinants of health from a life course perspective. I know several groups are very actively working on that. Our colleagues in University of Wisconsin have a multi ADRC study that's going to try to connect in a very standard way the social, economic, and also other social determinants of health parameters. We certainly need to have more comprehensive statistical models that's going to examine the potential modifying effect of social determinants of health. We need longitudinal data, which is very much missing in the picture here. And we need to go beyond the simple statistics of main effects. We need to look into the modification effects, the interactions. Now, I was awarded about 2 years ago at R01 to actually try to look, get into some of those issues I just presented. It's some study we call Sort Out AB, a study of ways to understand Alzheimer biomarkers. The specific aim is basically to determine cross-sectional and longitudinal racial differences in those markers, and also to determine, to assess modification by pretty much all the major covariates in this process. We are a multi-center study with WashU and UPenn, Emory, the ADRC, and also recently we did a revision to add the plasma biomarker to the consortium. We have UAB and the University of Wisconsin joining, in addition to Harvard Aging Brain Study, and also the baseline data of A4 trial. I don't think I have time, so I'm not going to talk about this, but what I really want to show is something, some picture we perhaps this picture will be good enough. We basically talk about the log ratio, hazard ratio between African American and Caucasian. You plot that ratio difference against all those possible important covariates. Baseline age is on the horizontal axis, E4 is on the top or bottom panel, and BMI, education, and sex. And you see a lot of different pictures. Okay, so this model using NAC UDS is talking about really on the CDR0 at baseline, how fast their progression to AD dementia. And the red line there is where things are different. The ratio difference is significantly different. So you can tell from this picture, it's a very complex structure in terms of the ratio differences. In our model, we found 4 to 6 significant three-way interactions in those analyses. And this picture incorporates all of those into the graphs. So I'm going to make a few concluding remarks, and then I will pay. So ratio differences may not be a simple binary answer, as this picture, the last picture clearly show. A much larger sample size would be needed to pinpoint the modifiers and even the direction. The picture shows in some domains, some areas, age or BMI or E4, some of those differences is in favor of African American. In the other cells, is in favor of the whites. So the statistical models must consider those interactive effects of all major AD risk factors. And the racial differences, of course, in AD biomarkers could have a significant consequences in the design and analysis of future prevention trials, because a lot of those trials are based on biomarkers. Finally, my acknowledgement to the participants, to our investigators at our center, and thank you all very much.
Thank you very much. Are there any quick questions? We have about 2 minutes. Okay, if not,